Good evening friends, welcome to SAM Biology class. Frederick Sanger's DNA sequencing method, also known as Dideoxy chain termination method, is a protocol that generates a nested set of DNA fragments by replicating a template DNA strand. Actually, the template DNA strand is to be sequenced. The nested set of fragments are obtained by interrupting the replication of template strand at different times by using unusual nucleotides known as dideoxynucleoside triphosphate. This is the general structure of a dideoxynucleoside triphosphate in which the hydroxyl group on the third carbon is absent. Due to the absence of hydroxyl group on the third carbon, when incorporated into the growing DNA strand, further formation of 3'-5' phosphodiester bond is prevented. As a result, the replication process is interrupted. So by using different types of dideoxynucleoside triphosphate, we can get a nested set of DNA fragments. Let us discuss about the protocol. This is the DNA template. We want to determine its sequence. In order to generate the nested set of DNA fragments, the DNA template has to be replicated. Initiation of replication requires a short primer which is complementary to the 3' end of the DNA template. It is because of base complementarity, the primer will best pair with the 3' end. The function of the primer is to provide a free 3' hydroxyl group to which successive nucleoside triphosphate are added one after another by DNA polymerase. But the problem is we don't know the sequence of the DNA template. Without knowing the sequence, it is not possible to develop the primer. Then how to initiate the replication? This problem is overcome by joining a short DNA strand of known sequence to the 3' end of the DNA template. Because we know the sequence of the short DNA strand, so we can develop a primer which is annealed to the 3' end. After annealing the primer, replication can be initiated by DNA polymerase. This problem can also be overcome by cloning the DNA insert in M13 phase. In M13 phase, the DNA insert which has to be sequenced is flanked by a non-M13 sequence. On the basis of this M13 sequence, a standard primer is developed and is annealed to this 3' end. After annealing the primer, DNA replication is initiated by including DNA polymerase 1 and 4 different types of deoxyribonucleoside triphosphates in the reaction mixture. DNA polymerase 1 extends the primer by adding successive nucleotides to the 3' hydroxyl group in 5' to 3' direction. The base sequence of the growing DNA strand is complementary to the base sequence of the DNA template. In this manner, replication will proceed in 5' to 3' direction. Then how to generate the nested set of DNA fragments? This is achieved by interrupting replication at different points of time. DNA replication can be interrupted by including dideoxynucleoside triphosphate in the reaction mixture. Dideoxynucleoside triphosphate do not have a hydroxyl group on the third carbon. It is due to the absence of the hydroxyl group on third carbon during the process of replication when a dideoxynucleoside triphosphate is added to the growing DNA strand, DNA replication is interrupted. In this example, we have added dideoxyadenosine triphosphate in the reaction mixture. During the process of replication, when a dideoxyadenosine triphosphate is added to the growing DNA strand, DNA replication is interrupted. As a result, a short DNA strand is obtained. Similarly, during replication, when another dideoxyadenosine triphosphate is added to the growing DNA strand at a different position, another DNA strand is obtained. So, by including 4 different types of dideoxyribonucleoside triphosphates along with DNA polymerase 1 and 4 types of deoxyribonucleoside triphosphate, 4 sets of DNA fragments are obtained. All these 4 sets of fragments together constitute the nested set of fragments from which the sequence of the DNA template can be determined. This figure outlines DNA sequencing by dideoxy chain termination method. This is the DNA template which has been cloned in M13 phase. Due to cloning, we have a large number of copies. This is the known M13 sequence based on which a standard primer is developed and is annealed to the 3' end. After annealing the primer, replication is carried out in 4 different chemical reactions in order to generate 4 different sets of labeled DNA fragments. This is achieved by dividing the copies of the DNA template into 4 different batches. In first batch, in the reaction mixture, 4 types of deoxyribonucleoside triphosphate and only labeled dideoxyadenosine triphosphate are included. In batch 2, the reaction mixture also contains 4 types of deoxyribonucleoside triphosphate plus only labeled dideoxycytidine triphosphate. In batch 3 reaction mixture, 4 types of deoxyribonucleoside triphosphate plus only labeled dideoxyguanosine triphosphate are included. And the last batch chemical reaction mixture contains 4 types of deoxyribonucleoside triphosphate plus only labeled dideoxythymidine triphosphate. In addition, the reaction mixtures also contain many copies of the DNA template. After this, replication is carried out in 4 different batches by employing DNA polymerase 1 and the same standard primer. These red colored strands are the DNA primer whose function is to provide a free 3' hydroxyl group to which nucleotides are added by DNA polymerase 1. Let us discuss about the fragments that are obtained from batch 1 chemical reaction. In batch 1, the reaction mixture contains 4 types of deoxyribonucleoside triphosphate plus dideoxyadenosine triphosphate. Dideoxyadenosine triphosphate do not have a hydroxyl group on 3rd carbon. It is due to the absence of this hydroxyl group during the process of replication when dideoxyadenosine triphosphate is added to the growing DNA strand, replication is interrupted. It is due to the random incorporation of dideoxyadenosine triphosphate at different positions. A number of DNA fragments are obtained, all of which have dideoxyadenosine triphosphate at the 3' end. Similarly, all the fragments that are obtained from batch 2 chemical reactions will have deoxycytidine triphosphate at the 3' end. This is due to the presence of dideoxycytidine triphosphate in the reaction mixture. But in base 3 chemical reaction, dideoxyguanosine triphosphate are included, because of which replication will be interrupted when a dideoxyguanosine triphosphate is added to the growing DNA strand. As a result, 3 different DNA fragments will be obtained, all of which have dideoxyguanosine triphosphate at the 3' end. But in base 4 chemical reaction, due to the inclusion of dideoxythymidine triphosphate in the reaction mixture, replication will be terminated at dideoxythymidine triphosphate. So, from these 4 different types of chemical reactions, we will get 4 different sets of labeled DNA fragments. After generation of different types of labeled DNA fragments, they are separated by electrophoresis by loading into a polyacrylamide gel. The DNA fragments, because of differences in their molecular size, will migrate at different rate under the influence of the electric field. As a result, they will be separated into different bands. This is a block of polyacrylamide gel which has 4 separate lengths. By electrophoresis, fragments of different sizes are separated into 13 different bands. The bands which are present in the first lane contain DNA fragments of different sizes, all of which have dideoxyadenosine triphosphate at the 3' end. It is based on the molecular size. The shortest fragment migrate at a faster rate and form a separate band which is nearer to the bottom of the gel. The shortest fragment will be followed by other fragments, but at a different electrophoretic mobility. As a result, all 4 types of DNA fragments are separated into different bands. But in second lane, 3 different bands are obtained, and the fragments of all these bands will have dideoxycytidine triphosphate at the 3' end. In third lane, all the DNA fragments of different sizes that are present in 3 separate bands will terminate at dideoxyguanosine triphosphate at the 3' end. This is due to the presence of dideoxyguanosine triphosphate in the reaction mixture. But in the DNA bands of the 4th lane, all the fragments of different sizes will have dideoxythymidine triphosphate at the terminus. In third lane, all the DNA fragments of different sizes that are present in 3 separate bands will terminate at dideoxyguanosine triphosphate at the 3' end. This is due to the presence of dideoxyguanosine triphosphate in the reaction mixture. But in the DNA bands of the 4th lane, all the fragments of different sizes will have dideoxythymidine triphosphate at the terminus. Because the bands are radioactive or they are fluorescent, these bands can be visualized by autoradiography or by fluorescence. Based on the relative position of the bands in 4 separate lanes, we can determine the sequence of the complementary strand which is read from bottom to top of the gel in 5' to 3' direction. Based on the sequence of the complementary strand, the sequence of the DNA strand can be determined. So friends, this is all about dideoxy chain termination method. For more updates, please subscribe my YouTube channel, Sam Biology Class.